Okay.
Okay. All right. Hi, everybody. I'm David, and I'm going to share some of my experiences doing exactly what you're doing, except with students. So here's just this slide. I always have to count on my fingers how many years I've been teaching, but it's something like that. I think this is going to be my 15th year, and definitely 10, so it'll be my 10th year doing the full sequence. For some reason, I started out the first time I ever did it with the full sequence, and I've pretty much done that ever since. At the school I teach at, which is the STEM Academy of Hollywood, which is on the Helen Bernstein campus, which is situated snugly between the Netflix campus and Home Depot. And it's funny, our school was built in 2008, but we have like track and field from late 1960s Texas, like right in the middle for some reason. We didn't import it, I guess we just wanted to emulate, but that's another story. So it's a pilot school, but we're also linked learning. We have two pathways, engineering and biomedical. So students select which pathway they want from their freshman year and follow that through graduation. And I say that because the students in the medical pathway do a competitor's version of bacterial transformation, which doesn't start as early in the process and doesn't go as far in the process, but they are familiar with a lot of the language that's involved in doing bacterial transformation and some of the concepts. But like I always tell them, okay, you did this one time. Does doing something one time make you an expert at it? I have one daughter. I'm by no means an expert father, although I tell her that. But so anyways, they still have a lot to learn, and oftentimes having that additional contact with the material can be helpful. So, but yeah, like I said, nobody, regardless of the pathway they're in, has gotten to Lab 6, which is protein purification, which is one of my personal favorites. Oh, did I click too much? Okay, so one of the things that I have my students do about a month or so before we actually get our Amgen kit and supplies is go through the Amgen sequence through Lab Exchange. Can I get a show of hands of anyone who's used Lab Exchange for anything? A couple. Has anyone used Lab Exchange for the Amgen material? Okay, cool. So I find it pretty helpful because it does take you through all of the steps without sort of the time sensitivity of doing it in real life, so to speak. Like, you don't have to worry about something incubating for too long or not long enough, or something not being cold or the right temperature, because it's all obviously in a digital virtual environment. And I've found that as much as I try to explain and scaffold and review, once we start the sequence, you only have 3 weeks to get it done. And some students get through the whole thing and their head is still spinning and they wind up missing the forest for the trees. And Lab Exchange is, I find, a great resource and tool for preventing that from happening, because you can go at a much more sort of, I don't know, slower pace, and students have time to grapple with the material and explore it the way they do. And this is sort of how it builds up when students log into Lab Exchange and they join your class. It kind of works like most LMSs or learning management systems, whether you use Schoology or Google Classroom or one of the other million ones out there. Same thing, there's like a class code, they join it, you've preloaded the sequence. Now, you don't have to reveal everything at once. You can kind of have things pop up as you go, which is what I do to kind of keep people on the same page and prevent them from getting too overwhelmed. So there's 11 modules in total for the whole Amgen sequence, from start, background information, video vignettes, all the way to protein purification. And I think the video simulations are great. And I'm not I grew up in the burgeoning age of video games, and it was always par for the course where you would always select the most difficult level. It's like, yeah, I played it for the first time, I'm an expert, and then get completely killed in whatever game you're playing. But that was how you learned. With these, don't be a hero. There's nothing wrong with starting at level 1, even though there's 3 levels to choose from, because level 3, it's almost like doing the real thing. Like, you have to be really precise. Your micropipette has to be directly lined up over your microfuge tube, and you got to close that, and you got to close the centrifuge, and it calls you out on every little detail, which is cool if you really want to put yourself in that realm, but also some of those details can kind of get in the way of forward progress. So play with them all, but there's no shame in doing level 1, because you get kind of the same experience and the same material as well. Yeah, so that is what I just said. Another cool thing about Lab Exchange, for those of you who haven't used it, is that it keeps track of everybody's progress through it. Not only does it tell you the percentage of the material that they've done, but the percentage of sort of the checkpoints and quizzes that they've gotten correct. So it lets you know, especially if you assign things asynchronously or as homework, who's doing what they're supposed to be doing. If they completed everything, but then they got like a really low percent correct, then they might just be randomly clicking through things to get to the end, and not spending much time. Oops, what happened here? On the questions and understanding the material. So it's a great way to check in with your students and keep track of what they're getting out of it. Also, while we're doing it, I afford a good amount of class time for doing it as well. I don't know how it is at your school, but homework completion rate at mine is not stellar. So knowing that, I try to reinforce this material so that I know a good portion of it is getting done, because that's sort of the point. So we use Schoology, and when I set it up in Schoology, I have, there's the learning target. So we used to have essential knowledge and skills, which were units, and now we use the term learning target, which are the same units. But anyway, so now we're on to learning targets. But within that, genetics and information transfer, there's an Amgen folder, which basically has the student lab protocol, this guy right here, any other materials they might need, visual learning guides, things like that. Then they have a separate folder for the lab exchange material, which I just showed you, and grades to keep track of that. Plus, those DNA transcription, translation, those are all links to videos, computer animations kind of showing those processes. And that's generally how I set up this section of the course. And they have basically their core materials and then other supported materials that we usually go over in class, but then they have access to at any time. So I've rattled off a good amount of information and facts. Do we have any questions that have come up, except for not being able to see around this pillar? I'm happy to take a pause for any questions if there are any. Well, yes, and
okay.
Yeah.
So I know the lab exchange has assessments, right? Do you take those assessments and input them into Schoology, or how do you use the lab exchange for assessment?
So I kind of keep them as is, but then I factor how students do on those assessments into their grade for lab exchange, if that makes sense. So I don't extract them, and I mean you could do that for sure, but since it's already in there and it is essentially graded automatically, I just leave it there.
So basically, I guess what I'm trying to ask is, do you create like an assignment separately on Schoology, and let's say, and you give them, let's say, lab 1.1 and they do a quiz on it, and let's say they get 80%, do you go like, oh, Johnny has 80% and you transfer it into Schoology gradebook, or do you just
Yeah,
not use
so
it at all?
let me see, do I answer that here? Okay, yeah. So each lab exchange module, I make its own assignment.
Okay.
And so I give them a grade for each one that they do. And then the rubric is here. So basically I'm looking for 90% complete and 90% correct because we're on a 4-point grade scale. And you know, this might seem pretty harsh, like less than 70% and less than 60% is only 1. But like I said, this is AP Biology. They have ample time to do it. Plus, there's plenty of time in class to do it as well. So this is just sort of, this isn't even you're not understanding it. This is just you're not doing it at that point. But does that answer?
Yeah.
Yeah. Okay.
Thank you.
And you had a question too?
I was just gonna ask, do you notice anything where like a student could maybe like cheat on it somehow? Like are they able to like ever like log in and like it like I don't know, like to where it would like shows complete but maybe they didn't like answer the question properly? Well, because that one they don't type in their questions, huh?
No, I mean I think it's multiple choice.
It's just like to watch it, huh?
Yeah, well there's, so there's, a lot of it is
Like
sort of a must complete, like
Yeah.
the assessments don't open up until you've done the pieces before that.
I guess my question is kind of like, what are some issues that you've seen with, if any, that you've seen that you've come up with that students are doing that could be like advice for us to like, when we set them up?
Well, so one thing I do is there is a good portion of this I do with them in class. So that way I'm, you know, kind of monitoring firsthand. I also don't give them a ton of time to just let it sit there and linger. Like they have to get it done by next class, or they lose their opportunity to get a grade for that particular portion of it. But I mean, the bigger issue of cheating, obviously, is huge, and I found it. You know, I struggle with it in terms of, you know, released question practice tests, homework assignments, things like that, and that's where, you know, we as teachers have to get creative as well. You know, whether it's forcing more handwritten materials during class time or what. I mean, that may be a topic for another chat. But there is one, we used Albert. Are you familiar with Albert? Albert.io. It's another sort of service for a lot of, well, any materials, but a lot of AP materials as well. And they have a cool feature where if you assign something, you can not only see how much they got correct, but how much time they spent on each question. And so you could, it became really obvious who was coming by their answers honestly and maybe not. It's like, you know, this kid got half of them right and spent 45 minutes. You got them all right and spent 2.5 minutes. So I'm not questioning your intelligence, but so this doesn't really have that feature. But for Lab Exchange in particular, because the grade is a combination of both completion and accuracy, and the questions aren't, they're not really meant to be super difficult. It's more of a review. I haven't had that be much of an issue.
So everything's been like running smooth?
Relatively,
Yeah.
relatively, yeah. Okay, So modules 1 through 5. I just listed everything, even though it all looks the same. Okay, so these are from your Amgen BioExperience Google Drives, and they fairly recently have put out these super helpful checklists and visual guides, which this is great when you're going over material, when you're going through the labs at the time that you're doing them. But these checklists are great supplementary material. They're very clear. It's, as long as you understand what each step is asking you to do, it's a wonderful sort of condensed version of this. And a lot of the students, especially students who get caught up in long chunks of directions, or just get intimidated by several pages of something to go through, these are great, and just a really helpful addition for that. And also, there's something that you yourself can print out if you have time. Make laminated copies. They're easy to store. They're all like 1 or 2 pages. So it's something that, obviously the PDF version of this is available for everyone, but not everyone may have a printed copy. So these can be a nice alternative for that. Then the visual guides, they're not necessarily checklists, but they kind of provide a summary, like kind of give a why for the what, with illustrations and diagrams that I think really help explain some of the more technical details of the labs. Like, for instance, for protein purification, it goes over the hydrophobic, hydrophilic interactions, the different buffers, what's happening to the protein, the different levels of protein structures from primary to secondary to tertiary. And again, correct me if I'm wrong, but I believe these are readily available for everyone. And there's one for each lab, and they're one page, and it's just sort of a great, like, okay, we're going to do the lab. Here's what we need to know, and we've completed the lab. Here's what I hope you have taken away from it. And they are visually, I think they're really well done, and they condense a lot of the information in one concentrated area, which again, helpful during a lab setting, and helpful for a lot of students who, might not be able to get through a lot of reading material efficiently. So, you had asked if I take the questions out of Lab Exchange and make that separate assignments. I don't do that, but I do take them out of this. And so I've gone through, the different chapters and labs, and over time, extracted the questions that I've found most salient and that there's not a whole lot of overlap with them, and I give these as assignments. And so this is like one document with all of the questions, but then I've also broken it up based on the chapter. And all of these questions are taken from the student lab manual throughout. So there's different, stop and think questions and review questions at the end of each lab or chapter. And so I've selected the ones that I think best represent the learning goals that I want my students to get, and I've put them together into these assignments. And yeah, that green doesn't look great on the presentation, but it's fine on your computer screen. And then here's a sort of the rubric that goes along with it as well. And then this is what it looks like just as a standalone assignment. I always have a rubric for all the assignments so the students know what they're shooting for, and if the students have an issue with the grade or the evaluation they got, I can always refer them to that and say, see that table that you didn't read? This is why you got what you got. But yeah, so, and I believe, so I'm not sure if the slides are being shared with you, but if they are, there's links to these documents for you to use, change, whatever, for your own purposes. And this is what it looks like in Schoology. So all of those questions are in the Amgen lab materials, and then it's basically 1 grade per each assignment. So another thing I do to increase accountability is students work in groups, which I think is probably the case for most of you, or will be. So I expect that they're going to work on the answers to these questions together, but I require everyone to submit their own copy. Now, it might seem silly because if they're doing it on a Google Doc, they can share it, and each one of them can just submit the exact same thing, and that's fine. But what ends up happening is, if only one person submits for the group, then in some cases, unfortunately, that means that's the only person who spent any time at all with those questions. By at least forcing everybody to submit their own copy, they have to take some responsibility for the work. And you can take it a step further and have groups individually assign different questions, and that almost always happens naturally anyway, because no one person wants to have to do all the questions for Chapter 5 on their own. So that usually ends up happening. So I also, as mentioned on the side here, post links to both Lab 4, Lab 5, and Lab 6 results, so that students can see how their results compare to the other groups. So, you know, maybe their ligated products didn't show up in their well, or their DNA marker ladder got kind of blurred during the process. So, you know, those things happen. But when you get to kind of compare your results with other groups, and obviously there's the models that are presented by Amgen in the presentations and slides that are provided, but being able to see what other students did, I think is really helpful, because they know that, okay, these are my classmates, they sit right over there at table 2, they got this, I know that they're not, you know, professional scientists somewhere doing this, so that's encouraging to me. So and plus that's part of the scientific process, right? Sharing your work, peer review, discussing it. So I try to make sure that component gets in there as well. And there's a lab 5. So the check for understanding is basically like a quiz/test, which again draws from a lot of those separate chapter questions, and either I use same questions or sort of adapted ones, and that's something that each individual has to do sort of toward the end. So in addition to the lab exchange, the questions, doing the lab itself, completing the work, this is also sort of like, okay, so you did all of that. What'd you get out of it? What do you remember? In your mind, what was the point? So this is a good tool for me to see where the students are at once they finally get to the end of it. Again, so these would be the student protocol checklist. These are created by Amgen, and the visual guides are also created by, or the Amgen Biotech Experience. And then these are the questions that are in here, but that I kind of combined into my own version of them as well. Okay, so these are, I added one more tip that I'll talk to you about too. But these are sort of tips and tricks that I've come up with over time. Always thaw your comp cells in ice, crushed, or whatever you have, right before aliquoting. You don't want to do it too early because you risk them no longer being viable, and you don't want to try to aliquot while they're still frozen, because then you might get really unequal distribution of cells because some might still be clumped together. So that's really important. For lab 6, if you do it, column equilibrating buffer is super important before you start, because it kind of goes through the resin column, and if this doesn't make sense, it will when you get to it, and kind of clears that out of any isopropyl alcohol or ethanol that might be in there to keep things from drying out. Because if you don't do that and you just start doing the protein purification with the ethanol in there, it immediately dilutes the protein, and instead of like this more defined pink band, it becomes sort of spread out already, and then you have to kind of convince the students that the product they got at the end really is more concentrated and brighter than what they started with, even though you all know that it isn't. So CEB, column equilibrating buffer, is really your friend in terms of that. Yeah, write down your own tips and tricks as you go. This I've had for years. It's got all kinds of notes and things like that. And at the beginning of the year, after my room has been cleaned and things are no longer where I left them, I'm always in a panic to find it again, because I feel like I would be really starting that at the beginning if I didn't have my trusty Post-it filled protocol with me. So I find it very useful. And the other thing I wanted to point out is, as you're and you're going to hear lots of tips and tricks, as I'm sure you have already as you go through, in terms of time management and labeling tubes and aliquoting and getting your materials together. In addition to that, when you're actually planning out your execution and timeline of the sequence, be careful to note which labs require extended or overnight incubation. So for instance, if you're doing the complete sequence lab 3, the ligation, you got to let those tubes incubate overnight so that the ligation actually has time to occur. So lab 3 itself is fairly quick, right? It's combining things in a tube and centrifuging. That's most of it. But it's not just you can do that and then move straight to lab 4. That's going to sit overnight. In addition, lab 5, once you plate your cells, those have to incubate at least overnight for you to get growth. And then lab 6, there's an incubation portion of that as well, which can be overnight, but I think you can get away with not quite as long. But yeah, so those are things to keep an eye out for too when you're about to do your lab and you're trying to make the most of your 3 weeks. And if you're like me, we're on a block schedule, so that means we don't meet every day. We meet every other day. So even though our classes are 90 minutes apiece, which is great for actually doing the lab, we don't have many of those classes within a 3-week time span. So you have to be really mindful about when you're scheduling, what things you can combine in order to wrap things up by the time you have to return your materials and collect your results. So,
I have a question.
oh, that's it. No, I'm done.
Okay.
No, I'm kidding. Go ahead.
I have a question about thawing.
Oh, yeah.
So you were talking about how for the competent cells, we're thawing them on ice.
Yes.
But then in labs 2 and 3, we also had frozen stuff, like the K+ and all of those. Those don't thaw on ice. Those just thaw room temperature.
Well, none of those things are living, right?
Okay.
So that's
Okay.
genetic material.
So that can
but
thaw in room temperature.
Yes. Yes.
Yes.
Okay.
Okay.
Yeah. So the comp cells, those are alive, and the temperature with that is key to not only preserving them, but making sure they're going to be viable by the time they get to your petri dishes. Yeah.
So you said crushed ice. Is there are there things like that, other things like that you recommend? So like we probably need a crushed ice machine?
Well, crushed ice is great because it increases the surface area of contact. I don't have that. I've asked for that. I have, let's just say there's no more discussion about that at my school, but the cafeteria has a great ice machine, and I've become friends with the cafeteria manager, and I can get plenty of that. A styrofoam cup, the ice cubes, and water as well is what I've been using.
Joe will cover all this tomorrow with
Okay.
transportation.
Okay.
Don't worry, we've thought of ways for you to do it.
Yeah.
Well, I just wanna see like how much, like what materials maybe I need to get ahead of time. Like I'm assuming I'm gonna need a fridge now, maybe a freezer, so I just,
Microwave.
yeah, microwave, so just thinking of things that I might need in my classroom or close by.
Yeah, I'll actually list the things you need to have on Thursday.
Yeah, a designated science microwave is really helpful for gels too. Yeah.
How do you feel like this fits into your pacing before the AP test? Like,
I mean, so I like doing it in late fall. I don't like to do it in the spring or spring semester because that's when I feel like a lot of AP teachers start to panic a little bit, like, oh, the test is in May and we still have to cover this, and this. So I like having this in the fall. It fits nicely with genetic information and information transfer, that sort of big idea in AP biology. It goes into depth on transcription, replication and translation, central dogma. That said, it's a lot of information. It's definitely a favorite of the test makers come May. So one of the drawbacks of doing it in November is by the time you get to the test in May, despite your best efforts and constant reminders, not everyone has been reviewing on a nightly basis. So it does warrant some review by the time you
so
get there.
you push Unit 6 forward?
Say again?
Do you push Unit 6 into the fall?
Yeah.
And push some other unit back?
Mhm.
What's
So-
your sequence then? For AP Bio?
For AP Bio?
If you push that to the late fall?
Well, just sort of in general
Yeah.
in general terms, I do that, and then I focus more on the evolution and ecology
Mhm.
sort of portions in the spring, and population genetics
In the spring.
as well.
So you push genetics to the back? Because it's like...
Well, I mean, yeah, I don't spend a lot of time on Punnett Square, so to speak, or Hardy-Weinberg. That I do in the spring more. But that said, this covers material that they should learn regardless. So even if when you get your materials doesn't precisely line up as to when you'd like to teach things, I still think you can make it work, and build around it if you need to.
In your experience, what's the most challenging lab technique or protocol for the students?
Probably 5. 5 first, and then maybe 4, just because 4, obviously you're dealing with teeny tiny amounts, you're aliquoting into the gels, you're needing to exercise patience as your gels run, and Lab 5 is very technical. There's a lot of steps. It's time sensitive. It's temperature sensitive. It's really cool, I think, because students at that point they know that this is sort of the culmination of what they've done, and they're going to be able to find out if the bacteria actually were transformed or not. But there's a lot going on, and it's like, okay, take them out of the ice, put them in the heat shock, take them out of the heat shock, put them back on ice. Don't only touch it by the rim and put it in this, and that goes in that container, and this is now in the autoclave bag. So there's, like I said, there's a lot of moving parts to Lab 5, so it's you know, it's a lot for the students to keep straight. It's a lot for you as the teacher to manage as well. But it is really cool. It's super engaging. There's very little opportunity to check your like updates on your phone. So I really like it for all those reasons, for sure.
David, do you have block or 50-minute periods? I'm sorry.
We have block. I mean, we have, so Monday is like our stack day. So we have 50-minute class periods on Monday, but then we have our A days, Tuesday, Thursday, and our B day, Wednesday, Friday. And those are all 90 minutes.
So do you try and have lab 5 on a 90-minute day?
Yeah.
Yeah.
Yeah, definitely. Yeah, lab 5 for sure on a 90-minute day. Yeah,
David?
lab 6 too.
When would you do lab 5 during the 3-week period?
Usually the end of the second week. And then we have the last couple of days to do lab 6. And depending on results, hopefully we get good results. We can, you know, loop a colony and make our own culture to purify for lab 6. If not, there's obviously other avenues to go. But that's usually the target. And you know, those of you who meet every day, your timeline's gonna be different, right? And depending on, I don't know, how your setup is, you may have to split labs. You may have to have students come in during lunch or after school to finish things up. And even I've had to do that for certain parts. But it in the end, it works. But again, lab exchange is helpful not only for the students, but you as a teacher too, because since really every single step is included in that, it's a review for you as well. And while students are going through that, it's another opportunity to refine your own planning process and make sure that you've got the things in place so that everything can get done based on the schedule that you have. What else?
David, when you started out, what labs did you do when you first started out?
Did them all.
I can't believe I let you do that.
I know. Me either.
I have a new rule, they can't do all of them.
That's probably good. That probably makes sense. But no one told me no, and so I did them all, and you know, I would say the first 2 years of doing it, it's not overwhelming like first day of school overwhelming. Like, who are these new people? Who are they? What kind of students are they going to be? But it is like, here we go again. But once you get into it, all that goes away because you're just in it, and the students are in it, and you're going through the sequence and you're getting it done. But this is why I say take your notes, because every time you do it, and I mean every time, you're gonna learn something. You're gonna come up with something that you didn't think of before that's gonna be super helpful, and you'll be like, yeah, this is great. And then you're done. For me, like, I'm done by winter break. If I don't write that down or make a note of it, come next Thanksgiving, I'm gonna be right back where I started. I'm not gonna remember. So do something where you take notes, because that's your process. That's you refining it, because the students are done. They don't need to know it, but you do. Yeah.
What were 2 things, or what are 2 things that you find to be the most helpful for students to be more successful in completing your sequence?
Well, I didn't always do the lab exchange material because it didn't exist. So I have found that students come away with a better understanding of what they're doing because of that. So I think it's just a really good learning tool, especially because students can go at their own pace. If a particular lab or simulation doesn't go well, it's not like, all right, that didn't go well, we got to move on anyways. They can go back and they can try it again. Where if lab 5 doesn't go well, let's try to figure out what went wrong. But more often than not, we don't have time to go back and do a whole nother transformation sequence so that you can learn from your mistakes. So Lab Exchange is sort of a great sandbox environment for that. And having them do those lab questions throughout, I think gives them time to reflect on what they did and process the steps that they've taken and the materials that they've used and what's really happening with their plasmids and why do these different pieces need to be there, and not only why do those pieces need to be there, but how do they relate to the overall central dogma? Like, how do they relate to how DNA is processed and created and copied throughout every organism? So I think having those built-in thought pauses are really important too. Yeah.
Okay, so this is something that occurred to me that I was considering, but because I've never done this before, but you've done it so many years, like maybe you can tell me if you think that's a good idea or not.
Okay.
So I did all the lab exchange, not all of it, I did like a lot of the lab exchange pathways that was assigned before this. And so I have a lot of EL students and a lot of students with different learning abilities. So that's always what's running in the back of my mind when I'm like imagining teaching scenarios, is like, how would these kids be able to understand this direction and be able to do this correctly, right? And also because I feel like labs like this is like, oh, if you screw up, it's like, like the consequences are quite huge. So I was doing the lab exchange simulation and I noticed that the simulation is basically identical as the actual lab, right?
Right.
And then I clicked level 1. Okay, and then so when I click level 1 and I do it, I notice that it is like, it basically tells you, like you were saying, exactly what to do. You have to close the lid before you do this. You have to eject the tip before they even let you go to the next one. So it walks you through everything, but then it is also at the same time making you do it. So it's not just, it's even better than having a diagram.
Yeah.
Because like you can move the stuff. So I was imagining, if I have a group of different ability learners or language learners, I can have someone that have their Chromebook out, and that can actually be their instruction step by step.
And use that as
where
the protocol?
one person is reading it, doing it virtually, and then this other person, okay, I did this, all right, then you do it here. And then like that kind of makes sure that they also doing it exactly correctly. So that's what I was kind of imagining, but I don't know if that is a good idea.
So what I would do, I think that's a great idea. However, I would also have a checklist too.
I would have that as well, yeah.
Because I'm not exactly sure if all of the measurements and amounts and temperatures and things
same.
in Lab Exchange are what they need to be. So
okay.
they might be, but
Just
these
in
for
case.
sure are.
Yeah.
But I think it's great for the action
Yeah.
of what you're supposed to
Yeah.
do. And when we say spread the contents from the P+ tube, this is what we mean.
Yeah.
I think it can be very helpful for that. I would just cross check it with the actual protocol.
Okay.
The temperatures and times are correct.
They are? Okay.
Yes.
Because they actually, you have to dial in the pipe
Yeah.
at the correct volume and
Oh,
Yes.
okay,
set
great.
the temperature
Yeah.
and the time and all of that.
Yeah.
Then as long as they understand, I guess, like the real life component matchup, like so, and I mean obviously the micropipette looks like the micropipette, but you might have a slightly different model.
Yeah.
Just just to make sure that they know how things match up.
Yeah,
I would say, but yeah, I might even try that myself. That's a good idea.
My one question would be, I'm worried about a Chromebook getting bacteria on it. Can you put it in a gallon Ziploc bag and still use it? Saran wrap maybe?
Oh, yeah.
Something like that?
Good.
I mean, that's why we have the lab protocol checklist, because we don't want them to have student guides that get things spilled on them and they're like, you know, they're either laminated or they're in a sheet protector, but I don't know. With the Chromebook, is that safe? to
You know,
have
you could
lab?
also project it and have it for everyone to see.
Yeah, but then if like every group is at a slightly different spot,
Mhm.
then that wouldn't work. But yeah, that's
Right.
a good thing to consider, yeah.
I actually
Yeah.
like that idea for when they're first doing the pipettes, and then maybe the next time seeing if they can do it.
Yeah.
But I like the idea of projecting it for their first time doing the pipette.
Yeah, no, that's a good idea. That could be really helpful. Cool. Other questions that people might have?
Do you have like an average kind of time for cleanup for certain labs, or which lab might need the longest cleanup, just?
Well, so anytime you're dealing with the live cultures and the cells, that's always more cleanup. Even if everything gets put in the proper waste receptacle, which never happens, but even if it does, you know, you're still having to wipe things down with your alcohol or percent bleach solution, and there's always, you know, some extra precautionary cleanup and cleaning you have to do with. I'd say lab 5 is probably the biggest culprit for extra cleaning. Lab 6 too, because you're dealing, but your lab 6, I feel like the culture is much more concentrated over less material, whereas lab 5, you're micropipetting, you're spreading, you've got you know, your petri dishes as well. So there's lots more points for possible contamination and things that need to be cleaned. Lab 6 a little bit less so, and then the other labs leaning up to are pretty easy. I mean, I think every step of the way, when you're dealing with more equipment, that it's more time to set up and more time to put away. But luckily, you know, when you get your supplies from your distribution center, it's pretty well organized. So you know where everything goes back to, and then you might even have your own setup in your classroom where, you know, this is where the gel electrophoresis materials are. This is where my incubator and shaker material is. So you can definitely do that. It kind of depends on how your classroom is set up, if other classes use your room when you're not there, so which is the case with me. So I got to make sure that everything is put away, or at least not within easy reach or sight. But yeah, so definitely when you're dealing with live cultures, you have to be a bit more fastidious about your cleaning. But you know, throughout, I require all my students to wear their PPE, gloves and goggles, and the gloves are discarded and the goggles are always cleaned and sterilized between labs. So that's, regardless, that's always part of it. So that's factored in. Yeah. So I don't know if any of my contact information is shared, but Amgen, you're welcome to. And hopefully, if anybody wants access to the slides, they'll give you that too. If for no other reason than you want to think through your own process and add to your own idea bank, that's what it's there for. Yeah.
In our lab exchange portion of the workshop, we encourage teachers to create their lab exchange accounts and friend request each other.
Oh, cool.
For ABE. Is that something we can do with you if the teachers create an account for them?
Yeah, I didn't even know you could do that.
Yeah.
I didn't know you could have friends on Lab Exchange.
If you label yourself an ABE, associated with ABE in your profile, then people can find you.
And you can use a forum to like chat amongst yourselves and ask questions.
Oh, that's great.
In general. So there is like a social media kind of aspect to lab exchange.
Yeah. Add me. Yeah.
I have another question. So do you only teach ABE bio?
No, I teach biology for freshmen
Do you
too.
do different ABE lab series with different classes?
Well, I only do the ABE labs with my AP Bio students. In part because we have multiple interdisciplinary projects with the freshmen that take up kind of a good portion of their fall and spring semester, which is probably when I would do it with them. So that's and that's a part of how our school is structured as a whole. But yeah, I only do this with my AP Bio students.
And then I just want to double check that you will send his slides.
I just with his permission, I just uploaded them to the drive folder that I've been updating for you guys.
Oh, okay.
So this PDI resources folder, it's there now.
Okay, thank you.
All right, so if there are no further questions, thanks for having me, and I hope some of what I said was helpful. And once we're all friends in the lab exchange world, we can chat and share whatever we come up with. So
Thank you so much.
thank you.